Hello, everybody. How are you all doing? I hope you all are doing great. Welcome to this session of Biotecnika. In today's session, we are going to talk about some Unit 8 important previous year questions, okay, along with a solution. So, as you all know, Unit 8 comprises of inheritance biology, which we also call as genetics. So, today I have taken the topic from where a lot of questions are asked, and especially we see more number of questions in the recent times. Okay, last 5 years paper, if you see, a lot of questions do come from this particular segment. So that is molecular markers and gene mapping. So whenever we see the questions, like there are some band patterns are given in the gel, and you have to comprehend it, and you have to link it with some or the other situation, whether it is linkage, whether it is recombination frequency, whether it is inheritance of a particular disease. So those kind of questions are becoming very common nowadays. Okay, so let's see some examples of those questions and see how we can solve them. So this is the question I have taken from June 2023 exam. Alleles capital A, small a, capital B, small b can be distinguished on the basis of their mobility on an agarose gel. The genes are present on the same chromosome. In the gel image below, the band pattern reflects the alleles in parents and their progeny. Number reflects the progeny counted. Which one of the following statements correctly explains the band pattern? Now, if you see so basically from the options that are given, we get an idea of what kind of, like in which direction we have to think. Okay. So if you see the options, it is talking about coupling configuration, repulsion configuration, A and B being in different linkage group. Okay. So first thing that we have to do is to coupling and repulsion configuration. So these are the different forms in which the alleles of a linked genes can be present. So first we have to find out whether the heterozygous parent is in which configuration the alleles are present. Okay. So heterozygous parent for sure is P1. How do we know that? Because in case of P2, we see only two bands. Okay. So one band for capital A, one band for capital B. Okay. Now, one more thing is very clearly mentioned in the question is that both the genes are present on the same chromosome. Okay. Now, since both the genes are present on the same chromosome, means they are part of the same linkage group. So, if they are part of same linkage group, then definitely option 3 you can cancel out. Okay, that they are in different linkage group. That is not possible because all the genes present on the same chromosome are part of the same linkage group. Okay. Now, what about P2? So, in P2, we see it has only two bands, capital A and capital B. Now, capital A and capital B is fine, and A and B are on the same chromosome. So, we know that on one of the chromosome, this is the configuration. Okay. So, we will first try to write it down in the linked gene format. Okay, so what about the other chromosome? Now, is P2 a haploid? Not possible. P2 has to be diploid. So, all the genes have to be present in two copies. Now, if we are getting only two bands, one for capital A, one for capital B, means that the other homologous chromosome of P2 is also having capital A and capital B. Okay, so from there it is clear that P2 is homozygous dominant for both the genes. Okay, great. Now for P1, okay, so we can also write down for P1, but then we don't know whether capital A will come with capital B or capital A will come with lowercase b. Okay, now to find it out, how we can do is, now this progeny we have to use. Okay, now A and B are present on the same chromosome, so they can be linked. And also when we check the progeny number that is given here, 75, 80, 22 and 16, we can see clearly that two two progenies are present in high number, whereas two progenies are present in comparatively lesser number. Okay, now what does that mean in terms of linked genes? That the progenies which are shown in higher number, they have to be the parentals. Okay, now what are parentals? Parentals means they will have the configuration that they have inherited from either P1 or P2. Okay, and what is this 22 and 16? These are the recombinants. So recombinants, how they are formed? Recombinants will be formed when recombination happens between the linked genes in the parents. Now from here you can find out the parental. Okay, see, when P1 cross with P2, okay, so this is P2. Now if I write down P1, okay, so when P1 is crossed with P2, we don't know about P1. So from P2, for sure one chromosome the offspring will get, which will be capital A, capital B, for sure. Okay, now if you check this class with 75, so capital A, capital B, you can eliminate that. For sure this is coming from P2. What about P1? So in case of P1, what is coming? So small b is 1, which is coming. So you can place small b. What about the other gene? So for A, there is only one band given, capital A, means both the homologous chromosomes have capital A only on it. So that gives us the idea, or that solves the question actually, that the chromosome that this progeny has received from P1 is for sure capital A, small b. Okay, so capital A, capital B coming from P2, capital A, small b is coming from P1. Now since it is parental, so there is no recombination is happening in the same form, it is coming from P1. Okay, so what does that mean? P1 has one chromosome with capital A, small b. So capital A, small b if you are placed, so the other chromosome will have what configuration? Small a, capital B. Okay, now this you can even confirm by this progeny, the other progeny with number 80. Okay, so that from there also you can find out that the chromosome will be small a, capital B. So when capital A, small b and small a, capital B is the configuration in P1, means it is present in which configuration? So this configuration is what we called as trans configuration or in repulsion. Okay, where the dominant allele of one gene is present with the recessive allele of the other gene. Okay, so this is called as repulsion or trans configuration. Okay, so from there what will be the answer? That in the heterozygous parent the alleles are present in repulsion configuration. So answer is option 2. Okay, very easy. You don't have to do any calculation. Usually students run away from calculation, but these questions, they usually do not involve any calculation, but definitely it involves the analysis. Okay, so you have to analyze the band pattern given, and then you have to link it with, here you have to link it with the concept of repulsion and coupling. Let's see another question, and this was from December 2022. Fertilization between two mating types P1 and P2 of Neurospora led to a diploid ascus cell, which gave rise to ascus containing 8 haploid ascospores. A set of DNA markers representing two linked loci was analyzed in P1, P2, and the octets labeled as 01 to 08, arranged from tip to the base of the ascus. The observed profile is represented below. Okay, so here we are talking about Neurospora. So Neurospora, it's a fungi. So it's a haploid fungi. So it will the spores will be formed. The spores will fuse and they will produce a diploid ascus cell. And that diploid ascus cell will first undergo meiosis, and then it will undergo one round of mitosis. Okay, which will result in the formation of 8 haploid ascospores. Okay. Right, now the markers have been labeled. So this is the pattern we are getting. Okay, so this is P1 and P2. Okay, and then these are the ascospores, the 8 ascospores. Which of the following is the correct conclusion of the above observations? Okay, so what are the options given? Band labeled A and C are allelic and segregation occurred during meiosis 2. Band labeled B and D are allelic and segregation occurred during meiosis 2. Bands labeled A and D are allelic and segregation occurred during meiosis 2. And bands labeled C and D are allelic and segregation occurred during meiosis 1. Now see here what we have to do. We have to link this band pattern to tetrad analysis. So if you see in one glimpse, this question does not appear to be of tetrad analysis. Okay, but since we are talking about Neurospora and we are talking about how the spores are arranged, so this is kind of tetrad analysis only we have to do on the basis of the profile which is given to us in the gel. Okay, right. Now first let's find out that which are the markers that are allelic. Okay, now what are alleles? Alleles are the alternate form of a particular gene. Okay, now since ascospores are haploid, so if there are allelic markers, so only one copy of that marker will be present. Okay, because they are haploid, they have only one set of chromosome, so only one allele can be present. Okay, maximum one allele only can be present. Okay, now if we see A and C, so like the spores, if you check out, right, so if if we see A and C, so the spores have either A or C. There is no spore which has both A and C. Okay, so they have either A or C, which means that A and C are allelic. Similarly, what about B and D? Now same in the same way. Okay, so you see spores with A or B and C or D, or A and D and C and D. Okay, but B and D, nothing. Not a single spore has both B and D. They have either B or D. Okay, which again means that B and D are allelic. Okay, so what we have found out, A and C are allelic, B and D are allelic. Okay, so so we can straight away eliminate option 3 and 4, because option 3 said that A and D are allelic. Option 4 is saying C and D are allelic. Okay, so these two are for sure wrong. Okay, 3 and 4 eliminated. Right. Now the next part is about the segregation of the alleles. Okay, now segregation of the alleles, if we have to talk about, so then you have to see the arrangement of the spores. Remember, so arrangement of the spores, how it will be? So when there is first division segregation, then what is the configuration we get? We get 4:4 configuration. Like if there is an ascus, so there will be 4 capital A's followed by 4 small a's. Right? That is how the arrangement will be. That is for first division segregation. What about second division segregation? So in case of second division segregation, two format can be there. One is 2:2:2:2:2. One is 2:4:2. Okay. So if it is 2:2:2:2 means 2 capital A, 2 small a, 2 capital A, 2 small a. This is how the arrangement will be. Okay. If it is 2:4:2 means 2 capital A, 4 small a, 2 capital A. Okay. Or the alternate arrangement can be 2 small a, 4 capital A, and again followed by 2 small a. Right? So these two are the arrangements which can be there in the second division segregation. Right? Now what are these bands? These bands are also alleles only. Okay, so if you just want to assign it according to A and capital A, so basically this can be the band for capital A, this can be the band for small a. Okay, so what kind of arrangement do we see here? 4:4. What about the other one? So if you consider, let's say if it's a different gene, so if I am giving it capital B, okay, the other one is small b, right? So now in this two, what kind of pattern we are saying? 2:2:2:2:2. Okay, so 2:2:2:2 means this for this gene B and D, which are alleles of each other. So here segregation is happening in the second division, whereas in the first case, since the pattern is 4:4, means segregation is happening in the first division. Right? So it is very, very easy. Is it not? Just you have to link it. Okay. So band pattern is given, but you have to link it with your basic understanding of tetrad analysis. Okay. So what will be the answer? So band B and D are allelic and segregation is during meiosis 2. So answer will be option 2. So option 1 is wrong, because for A and C, segregation is happening during meiosis 1 and not 2. Alright. So similar band pattern only given, but what is the concept that you have to apply is different. Let's see one more question. Okay, so here also again band pattern only given. Okay, so double haploids are plants derived from the single immature pollen and doubled artificially to form diploids. A double haploid population was created from F1 progeny derived from a cross between two parents P1 and P2, one resistant capital R and the other sensitive capital S to white rust. In parents F1 and DH population were profiled with a set of codominant marker which is represented below. The following table summarizes the proposed percentage of the four different types 1 to 4 of DH progeny, assuming that the DNA marker is first unlinked to the gene governing the trait and second linked at a distance of 10 centimorgan. Which one of the following options correctly represents the expected ratio of unlinked and linked respectively? Okay, so again, so for the first part, okay, so first part is quite easy. So here, the first one is saying that what will be the situation if the gene governing the trait and the molecular marker are unlinked to each other. Okay, so so basically instead of molecular marker, just think of it as two genes. So if two genes are unlinked, means they are assorting independently. So if they are assorting independently, okay, then in the cross, what do we expect? Yes, we have to apply Mendel's third law, that is the law of independent assortment. So if two different genes are getting assorted independently, then in the next generation, we will expect all the possible combinations in equal proportion. Right? So if there are four possible combinations are there, so they all will be present in equal proportion. So each will be 25% right? So there are four varieties, so each will be 25%. Okay, so unlinked means independent assortment, and that is from there we got it. So for unlinked, answer will be that in profile A. Okay, that is 25, 25, 25, 25. Second one is asking what if the marker and the gene is linked at a distance of 10 centimorgan. Okay, now for this let's do some basic calculation. So when distance is given as 10 centimorgan, what does that mean? That recombination frequency percentage is equal to 10, right? So if recombination frequency percentage is equal to 10, again what does it mean? That number of recombinants divided by total progeny will be equal to 10%. Right? Now there will be two recombinant varieties and there will be two parental varieties. Okay. So what will be the proportion of one recombinant variety? So proportion of recombinant 1 will be equal. Proportion of recombinant 1 will be equal to proportion of recombinant 2. And how much it will be? Half of 10%. Okay. So it will be 5% each. Okay. So if recombinants are 5% each, then what about the parentals? Okay. So in this case, parentals, I am writing down here. So parentals again will be 2, right? So total parentals will be equal to 100 minus 10, that is 90%. Okay, and proportion of parental 1 will be equal to proportion of parental 2, which will be equal to 90 by 2, that is 45%. Okay, so parentals will be 45% and the recombinants will be 5%. Okay, now if you see in the table, so definitely you can eliminate 3. Okay, that is not possible. So it will be either 1 or 2. Now for that, what you have to find out? You have to find out that who are the recombinants and who are the parentals. Okay, so parentals will be what? So in DH population, parentals will be the one who are exactly matching either P1 or P2. Okay, so they have to match not only in the band pattern, they also have to match whether it is resistant or sensitive. to the white rust. Okay, so when we do that, so this one, the 1 is exactly matching P1. Okay, it is RR and these two bands are there. For P2, if you see, it is exactly matching this category 4, where it is S, S and the band pattern. Okay, so that means this one and this one, they are parentals. Okay, and I'm just coloring the other two with blue. So this one and this one, they are recombinants. Okay, so recombinants are expected in 5% each, parentals are expected in 45% each. So what will be the profile that we'll see here? So it will be first parental, so 45%. Then there are two recombinants, 5%, 5%, and the fourth group will be 45%. Okay, so if you see it, so answer will be option 1. Okay, not option 1, from the table. For the linked, the profile we will expect is for 1. Okay, so answer here will be option 2, that is A for unlinked, and the possibility, the profile of 1 for linked. Okay, A and 1 will be the answer. All right, so these are 3 different varieties in which the questions are asked. Okay, given you a band profile and how you have to link it with different concepts related to gene mapping. There are a lot of other questions like this. I would request you all to try them. Just check out the previous year question paper and try to solve them. And if you are not able to solve, then please write to us in Biotecnika and definitely we will make a video out of it. Stay tuned for more such previous year question discussions in our YouTube channel. Thank you everyone. Bye-bye. Have a great day.